Hello, everyone, and welcome to Nature Nano Talks. I'm Chiara Pastore from Nature Nano, and we have the pleasure to have with us today Professor Robert Parton from University of Queensland in Brisbane, Australia. He will chat with us a little bit about his review on endocytic mechanism and pathways for cellular uptake of biological and nanoparticle therapeutics. So thank you very much, Professor Parton, for being with us today.
Thank you for inviting me.
So to start with our conversation, one general question that we have is, why did you decide to write such a piece, and what was the stimulus to write this article?
So my own background is in cell biology, particularly endocytosis, which I've worked in for a long time now. And I started to work with Angus Johnston, who's an expert in nanoparticles and drug delivery, and he's actually a co-author on this review. And when we started to look at papers on nanoparticle endocytosis, we were a little bit surprised that we felt many of those papers were not making a significant contribution to the field. And most of those studies were performed perhaps by chemists who were taking their nanoparticles. They were taking a standard cell line, adding the nanoparticles to the cell, and then they were seeing what the uptake pathways were for those particles. And we had serious questions about some of the conclusions that were being made from those studies, which generally involved chemical inhibitors, looking only at the endocytic uptake pathway rather than endpoints about the efficacy of the nanoparticles that they were using. And we felt we should maybe raise a kind of alarm that maybe we should reassess how these studies should be done. We should also provide an overview of the methods that can be used to study endocytosis now, which I think have advanced quite a long way, and the kind of misconceptions in the field at the moment. So that's what we're trying to do in this review.
Okay, I see. So you mentioned that the many papers you find surprising. So what was why were they so problematic? What really struck you there?
So one thing that we immediately noticed, my own expertise is in structures called caveoli. These are cell surface pits. They sit on the surface of the cell, and they've been implicated in the endocytosis of many nanoparticles. But we know a lot about these structures. We know how they form. We know that the proteins that are involved in their formation. And we were surprised to see that cell types, cell lines that are used in some of these studies, we know that they completely lack caveoli. They lack the major structural proteins of caveoli. And yet the conclusions of these papers were that caveoli were a major uptake pathway for these nanoparticles. So that was immediately a bit of a red flag to us. And we started looking at these papers, and we also realized that there are a number of inhibitors were being used to draw these conclusions. And looking at the papers, we felt that there was a lot of misconception about how specific these inhibitors were. And if I could actually show the kind of scheme, I think, for endocytosis.
Sure.
So this is a summary from the review of what we believe the major endocytic pathways in a cultured cell. So we have clathrin-mediated endocytosis. This is the major pathway that's involved in nutrient uptake. Then we have a fairly newly discovered pathway, FEAM. This is a fast endophilin-mediated endocytic pathway, and that's for ligands that are binding to receptors and then stimulating their uptake. We have the caveola pathway and the major protein caveolin that can serve as a marker for this pathway. Then we have the click-geek pathway. These are tubular carriers, and these are actually generated by extracellular ligands that can bind to receptors, bind to lipids on the cell surface, cluster those proteins, then induce this formation. And then at the side here, we have macropinocytosis, the engulfment of large particles, and then phagocyte of large amounts of fluid, I should say, and then phagocytosis, which is for the engulfment of particles. Mm-hmm. And we know that perturbation of, for example, cholesterol has been used as a way of perturbing caveoli, but in fact, perturbation of cholesterol will affect the FEM pathway, the caveoli pathway, and the click-geek pathway fairly specifically, in that the clathrin pathway under these conditions can still be working normally, but you can't use those kind of inhibitors to really show the endocytic pathway for these kind of with these kind of inhibitors. We also wanted to summarize the pathway because it's clear that only smaller particles are going to be able to be internalized through these pathways. So through the clathrin, the FEM, the caveola pathway, the click-geek pathway. The larger particles would have to be internalized by these other pathways because they physically wouldn't be able to get into these. Now, this seems fairly obvious, but in some of the articles, this wasn't really used as a criterion for deciding on the pathways that were being used. Another point we wanted to make was that there are now markers for these pathways. So there are ways to manipulate these pathways by genetic means. So this can mean, for example, knockout of caveolin or cavin for caveoli, endophilin family members for the FEM pathway, and various other manipulations that can be done, taking advantage of the molecular understanding of these pathways now. And I think we tried to bring that out in the review. Not just that the inhibitors are somewhat non-specific, but also that there are other means to modulate these pathways to understand what's going on. The other point we wanted to make in this review is really, if we are looking at these pathways, what we have to do is correlate the uptake via these pathways with an endpoint. So for many nanoparticles, they can be taken up into the cell, but the rate limiting step is that those nanoparticles need to get out of the endosome, the late endosome or the lysosome, and then reach their targets, which are often the cytosol. And an example of that are the vaccine particles that are made up of the mRNA plus lipids. They have to get from the outside of the cell into the endosome, then across a membrane into the cytosol for that mRNA to be translated. So really, unless you're correlating an endpoint or even the cytoplasmic delivery of a nanoparticle, then in a way, understanding the endocytic pathway is not as important. But once you know that you have a functional endpoint, you can now look which is the most important pathway into the cell. And then you can start to think about modulating the properties of the nanoparticles so that you can get your efficacy of the particle that you're trying to put in, but also you can then correlate that with endosomal escape and correlate that with increased endocytosis.
Okay, so looking at your slide, of course, there are several different pathways that you were saying, and then there is the issue of endosomal escape. So I'm wondering whether there is a correlation between a specific endocytic pathway and endosomal escape, and what can material scientists, for example, learn from understanding better endocytic pathways, and how can they use this information to design better nanoparticles?
Yeah, I think that's a great question, Chiara, and I think in many cases we haven't got a good correlation between, say, endosomal escape and a particular endocytic pathway. I think there are some studies that have started to do that, and I think this is really important, but I think in many cases there hasn't been a good correlation between a particular endocytic pathway and the endosomal escape. And it's interesting that on this scheme, I think the consensus now, the dogma is that all of these pathways, these diverse pathways, might be used to bring in different things into the cell, but they do all go to the early endosome. Some of that material would be transported to the same late endosome and then the lysosome. So really, whether you get the endosomal escape, does it really depend on the endocytic pathway, or is just increasing the endocytic uptake enough because a fraction of what you get into the cell is then going to be escaping into the endosome? And I think some studies have done a great job of looking at that, but in many cases we don't know. So I think there are two things. There's having assays that discriminate between uptake and endosomal escape, so you can start to correlate those two different processes. And The other point is that we need to understand these endocytic pathways to make sure that we get the maximum amount of a nanoparticle into the cell. And I think the click geek is an interesting one. This is, again, probably not so well characterized in some ways, but we do know that endogenous proteins can cross-link lipids and proteins on the surface, and that's also been exploited by toxins and also by viruses to induce their own entry into the cell. So you can see that if nanoparticles can be engineered in the right way, they can actually utilize those kind of pathways to get into the cell, and you can maximize the uptake of those particles. We have to think about the size of the particles. We can think about things like the corona. We can think about how soft or how hard the particles are. I think then we can start to correlate with these different pathways and start to understand how important those properties are for the initial endocytosis pathway, but also for the endosomal escape as well.
This is really interesting. Thank you. And so what are the key messages that you'd like to convey in the review? What's your take-home message in a way?
Okay, so I think the first one is that we need to think about the way the cell types that we use to study these pathways. We know that not all these pathways are in every cell type. And is it really relevant to the cell type that a researcher wants to target in vivo? So is it enough to take a cultured cell? It probably is in the initial stage of the project. But then do we need to think about in vivo models, zebrafish, mouse, organoids, so we can actually look at what happens in a more physiological context. And when we think of a nanoparticle in vivo, which might be injected into the bloodstream, it needs to cross the endothelium, it needs to then reach the target cell, and it needs to be internalized by that target cell. I think we also have to keep in mind that whenever we're doing these studies, I don't think it's sufficient to just say your nanoparticle is taken up by a certain pathway. I think we always have to be correlating that nanoparticle with an endpoint that might be reaching its target in the cytosol, it might be some other functional endpoint, so that we can then correlate the endocytosis that we're seeing with the actual efficacy of that nanoparticle you're looking at, because then we can start to modulate the endocytosis in a way which will help us to make those nanoparticles more effective. And so the third thing I'd say is that we need to think about the way we do these experiments, the inhibitors that we use. Inhibitors are fine as long as they're very carefully controlled, but there are other genetic means to modulate pathways now. There are knockout cells for many of these pathways. And I think we're also going to see in the future, we're going to see more acute methods of genetically knocking down proteins as well. And that might be very transient CRISPR-based methods. It may also be that we acutely degrade the proteins involved in these pathways so we can switch them off quickly and see what effect that has on the pathways. And I think, the final thing I'd say is that, just think carefully about what the meaning of the nanoparticle endocytosis experiments are. What's the endpoint? What's the goal of this? And what do you really want to see in vivo with your nanoparticles, so that we can make them more precise and we can make them more effective.
That's great. Thank you. So a lot of new avenues in a way to actually better understand this the mechanism. So that was really, really interesting and really informative. Thank you very much, Professor Parton, for joining us. It was really a pleasure to have you, and it was really good to learn more and to know more about how this issue can be tackled. Thank you so much again.
Thank you.